Okay, my smart learner, let us now look into the key concepts. Number one, the gene. What is a gene? You know that wena dark beauty ufuze ubaba, noma umhlophe ufuze uma, noma umude, noma umfushane, noma uyisidudla. Mhlambe awufuzanga uma, mhlambe awufuzanga ubaba kodwa ufuze ugogo. And mhlambe awufuzanga ugogo, mhlambe ufuze ugogo kagogo, noma ugogo kagogo kagogo. So it's all about the genes. So you know that our genes, they are transferred from generation to generation. They are inherited from generation to generation. Ayadlulela ufuzo. Mhlawumbe njengokuthi nje abantu bakabani baney'ndlebe ezithe, amehlo athe. So it's all about our genes. Okay, so then what is a gene? You know that in grade 10, you have learned about our cells, that in our bodies, we have cells. And in our cells, we have organelles. And in those organelles, there is an organelle called a nucleus. And in that nucleus, there are chromosomes. In that chromosome, there is something called a DNA that you have done in the first term. And in that DNA, there is something called a gene. So, a gene that we are talking about, the gene that are inherited from generation to generation. They've got a genetic material in them that is being transferred from one parent to the other. So, a gene is then a small portion of DNA coding for a particular characteristic. And then let us look at the second one, alleles. Now, what are alleles? Alleles are different forms of a gene which occur at the same position on a homologous chromosomes or at the same locus. It is important, my grade 12 learner, to understand these terms because then they are going to make you understand the whole genetic topic. So if you don't understand the terms, you'll find that in the questions, in your exam, in your test, there are terms that are there and then you will struggle to answer because then you will not understand what the meaning of the term. So it is important to first understand these concepts. So then an allele is a different form of a gene which occur at the same locus or same position or on an homologous chromosome. You also find these terms in multiple choices. So then it's very, very important, my dear grade 12 learner, to know them. So if you would look here, let me just get my pointer. Let's get a pointer so that you can see. Okay, here's my pointer. When you look at these homologous chromosomes, you will see that there is a label for alleles, the big T and the big T. So as you can see, they are at the same position. So that's a definition of an allele. Let's move to the next one. The genotype and the phenotype. Let me do this together at the same time because then learners tend to get confused. I've seen many learners getting confused between the two terms. They ask you about the genotype and then the learner would give a phenotype answer. My grade 12 learner, it is very important to differentiate between the two terms. So you know that in the alphabet, there is the first alphabet is A and the last alphabet is Z. So you know the A to Z, you know the order. So it's also important to also try and know how to differentiate between a genotype and a phenotype. So then, what is a genotype? Let's start here. The genetic composition, the makeup of an organism. Now, always remember, my dear grade 12 learner, that if you want to express a genotype, you would express it using the letters. So you know I'm a letters A to Z. So you would use the letters. For example, for a tall plant, if you want to express genotype for a tall plant, so uzoyi expressa uthini uthi big T or big T small t. That's a genotype. So you will notice you are using the letters, my dear grade 12. Don't get confused between the two letters. And then if you want to express a phenotype, then what is a phenotype? A phenotype is then a physical appearance of an organism. So njengoba une-plant, i-genotype yakhona big T small t noma big T, those are the letters. So then from big T small t, what would you get then? You would get a plant that is tall, or you would also get a plant that is short as well. If uno small t, and get. So then, if phenotype then is a physical appearance of an organism that is determined by the genotype. Usually, each genotype, the letter J comes first before a letter P. So that's how you would remember as well. And also, each genotype we express letters, letters A to Z. So we express letters. It's either o T, noma o R, noma o B, noma bani, any letter that is given appropriate to the question. So then each genotype you express it with the letters. Phenotype uniga igama. So ama-alphabet in alphabetic order A to Z. Uyazi ukuthi lawo ama-alphabet enza igama. So A banbani you can make it grade 12 angithi. So those alphabets. So then phenotype as well. Yenziwa yini genotype. So uno T big T wenza ini utho. That's a phenotype. Uyikhumbule ungayikhohlwa. Please don't get confused between the two. And then let's move to dominance. Into a dominance. That means into a boss. You know, when in your school you are dominant because when I grade 12 gets. So into a dominance. Into a kamaya kunenge. For example. So if uno big T small t, big T dominant, small t yini recessive. Let me just do them together as well. So the allo that is expressed shown in a phenotype when found in the heterozygous. So heterozygous as well isn't fine. Hetero means a different, homo means as the same. So then we dominant. If uno big T small t, that's heterozygous. And so then because big T is dominant, then that plant will be tall. And homozygous, homo means the same. Big T, big T. So njoba go big T, that's uti dominant. So then those plants will be tall. So I find at tall or big T small t or big T, they are all tall because then dominant. And then recessive is an opposite. So recessive, for example, small t. For example, if you look here, with dominant allo, big T is for tall plants, recessive allo, small t, and get to your bone. So then, if you would come here, we are going to go to a good genotype here, big T, then the plant is tall. If you come here, small t, then the plant is short. If you come here, big T small t, then the plant is tall and get to your bone. So then that's the difference between the two. There are learners, I don't want to take it for granted, but there are learners who still struggle between these terms. That's why I tell you that it is important before you master the topic of genetics to understand the key concept, because then if they ask you a certain question, you might find that you understand, you know the answer, but then you get confused between the terms. So that's why I thought you must go through the concepts first. And then heterozygous, I've explained heterozygous, it's not the same. Big T, small t is heterozygous. They're two different. It's also important to read at the definitions because it will help when it comes to terminology. 10 marks wonke my grade 12 learner on your final exam is terms. Wonke or sometimes 12 or maybe 15. So every mark counts over grade 12. So heterozygous is two different allos for a particular characteristic. So now we are born and ongafani, in other words. And then homozygous okfanayo, it could be T, big T, small t, small t. Let's look into this one, monohybrid cross. Now, a monohybrid cross, mono means one. Die, two, try, three, etc. So mono means one. So that means that a monohybrid cross is a cross that has to do with one characteristic. So then a monohybrid cross, only one characteristic or trait is being shown in the genetic cross. For example, a flower color, only, for example, kuvela yellow and white. So it's all about the color, whether yellow or white or pink or blue. It's all about the color. So it's showing one characteristic. Or maybe the shape. It's either round or wrinkled or skew or oval or whatever shape it is. So it's showing one characteristic. So then it's showing what? It's showing the shape. So it's either it's showing the color or it's showing the shape. So we call it a mono because then it's showing one characteristic. And then complete dominance. There are three types of dominance. Let me just get my pointer here so that I can just point whatever I'm showing. So it completes dominant. Now, a complete dominant is a genetic cross where the dominant alleles mask or they block the expression of a recessive allele. So usuyayazi a recessive allele in the heterozygous condition. Usuyayazi heterozygous because it's different things. So big T and a small t, for example, recessive small t is recessive. So then a complete dominant, it blocks it blocks the expression. For example, here, if I'm crossing two plants, the tall plant and a short plant, tall is big T, and a short is small t. Then you will get a plant that is tall because then big T and small t, and you get a plant that is tall. And by the way, tall actually is a phenotype, as I've told you. What's a phenotype? Is the physical appearance from each genotype. Each genotype is big T and big T. Each genotype here is small t and small t, and then you get a plant that is tall because then big T is dominant over small t. Small t is recessive. So then this is called a dominant dominance because then the one that is dominant is blocking, is masking the one that is recessive. That's a complete dominance, my grade 12 learner. And then you also get an incomplete dominance. So then, what is an incomplete dominance? You need to know the difference between the two. So let me just use the example again. If you would cross a flower that is red with a flower that is white, so you're crossing red and a white, and then you get a pink color. That shows that this is incomplete because then red no white, if you mixing them together, colors, if you mix these colors together and then you get a color which is then pink. So even with crossing of planting, you cross you cross a red flower and a white flower, and then you'll get a pink flower. We call that an incomplete dominant. And an incomplete dominant is a genetic cross between two phenotypical different parents produces offspring different from both parents, but with an intermediate phenotype. So then ingane oyitholayo ayifani noma ayifani nobaba, but ibabhangile, we call that an incomplete dominance. We do get such things. Uthola ukuthi ingane noma usisi wakho abantu badida hayi ngoba ufana nobaba ngoba ufana noma, because then ubaxube bobabili. For example, if a red flowered plant is crossed with a white flowered plant and there is incomplete dominant, the offspring will have a pink flowers that we call an intermediate color umbala. And the third one, the codominant. So usuyazi complete dominance, usuyazi incomplete dominance. Now let us look at the codominance. Codominance yona iyini. Let's again look at the example. So with codominance, you are crossing now a red flower and a white flower, and you get a flower with red and white patches. So then kuyabalansisa-ke awusatholi intermediate color, so you get both colors linganayo. So we call that a codominant. Siyabambisana, sithola into elinganayo, unlike complete incomplete. So codominant, you thola into elinganayo, izimbali ezimbili, red no white, ezophuma enoku red ibe no red ibe no white, just like inkomo. You do get inkomo ezimhlophe, uthola inkomo ezi-black. Then if uzocrossa lezo zinkomo, then uzothola inkomo enokumnyama nenkomo umhlophe. So we call that a codominant. A genetic cross in which both alleles are expressed equally in the phenotype. For example, if a red flowered plant is crossed with a white flowered plant and there is a codominant of offspring, has flowers with red and white patches. So it's kubalulekile bantu bami ukuthi to understand these three types of dominance. And then you go to multiple alleles. Okay, this has to do with the blood grouping. More than two alternative forms of a gene at the same time, same locus, I mean. For example, blood groups are controlled by three alleles, namely, big I to the power big A, big I to the power big E, and small I. So ama alleles are controlled like igazi out three. So we look into that. Many learners have seen even high flyers that struggle with when it comes to blood grouping, but you get them when you get them. Number 14, the sex-linked characteristic. We know that you have 23 pairs of chromosomes, and you know that chromosomes. So you've got sex chromosomes, and you know that as well. You have sex diseases, so you also know that you need to cross. This is very important. Many learners make mistakes here. Oh, it's so sad to mark a paper. Uthole ukuthi ingane understands the crossing A to Z, but then the learner confused with the sex-linked diseases. We'll get into that as well. So sex-linked characteristics, the characteristics or traits carried on the sex chromosome. For example, hemophilia and colorblindness. Angithi uyazi ukuthi akhona guidelines akho, they are there and you need to know them. The alleles for hemophilia or colorblindness are indicated as the superscript on the sex chromosomes. For example, you know that if you're a female, then you have two X's, and if you're a male, you would have you have X and a Y if you're a male. If you're a female, you have X and an X. So if you are normal as a female, then you have two X's and the BH, okay, is superscript. That's what you're talking about. So as you can see here, for example, or X about one gets as female. So go X okala you would have a big letter H. Now go X you would have a big letter H. That tells you that unjani you are normal. You can also be normal. Ube uno X and it's when we female. Uno X about 2. Uno X and as you can see here, big letter H and a small letter H. You are still normal because unalo H lo omkhulu. You are normal. But then if une hemophilia, uzobanjani nabo H two abanjani abancane, as you can see here. So when that means when unalesi sifo lesi. Now when it comes to a female, I need to a male, I mean. So email uyayazi ukuthi X and Y. So email yona izoba ne letter eyodwa. If normal izoba no big letter H. If in a hemophilia izoba no small letter. So it's easy email because izoba nama letter eyodwa, which makes it easier crossing which you guys want to remove hemophilia or not. But when it comes to solving, to crossing, because this is important to know. And then number 15 then will be karyotype. This is your karyotype. I'm going to ask you, you've got 23 pairs of chromosomes. In other words, you've got 46 chromosomes. Now, the number, shape, and arrangement of all chromosomes in the nucleus of a somatic cell. This is how it looks, and you know, sex chromosome wakho. Number 16 would be cloning, the process by which genetically identical organisms are formed using biotechnology. So you know ukuthi ungakwazi create an organism, and a cow has been cloned, a sheep has been cloned, I think a frog has been cloned as well, and a human being can be cloned, but it is illegal. Ungakwazi ukwenza umuntu, but then it is not legalized. So for example, the Dolly the sheep was cloned using a diploid cell from one parent. Therefore it had the identical genetic material of that parent. So cloning, that's all about cloning. Noma bengabuzi kakhulu, but it's important also to know it, as it's also in your guidelines. Then genetic modification, number 17, the manipulation of the genetic material of an organism to get a desired changes. So they're using these genes ukuthi benze noma yini abathanda ukuyenza, baclone benze umuntu, bashintshe benze. This question also came, I think it was in 2018, and ingane zami 5 marks wonke because the book we're using didn't have this, and didn't look at the guidelines. It's in the guideline. I think it was 5 marks wonke. The insertion of human insulin gene in plasmid of bacteria so that the bacteria produces a human insulin. Okay, it's important also to know this. So get into it when you get there. 18, the human genome, the mapping of the exact position of all the genes and all the chromosomes of a human. For example, gene number 3 on chromosome number 4 is responsible for a particular characteristic. Okay, so my grade 12 learner, it's important also to know this concept before we get into the topic. This is your activity. I want you to do this so long. These are just the 15 terms in genetic topic. I want you to answer them, and then when I come back to you, I'll be giving you the answers because it's important to understand the terms. And then after understanding the terms, then you'll get into the topic, starting with the next topic as shown you before. So I wish you good luck, all the best, goodbye.